for the conference organizing committee, and thanks for your attention. My name is Junjun Qiu. I'm from Obstetrics and Gynecology Hospital of Fudan University, Shanghai, China. The Valencia is a beautiful city. I'm very glad to be here to share my recent research with you. The title is Long Non-Coding RNA HOTAIR promotes the proliferation of serous ovarian cancer cells through the regulation of cell cycle arrest and apoptosis. I will describe this topic from the following aspects: the introduction, the materials and methods, results, discussion, and conclusion. Introduction. As we all know, ovarian cancer is highly lethal, and the most prevalent ovarian cancer histotype is serous ovarian cancer, which often presents at advanced stage and is often accompanied by malignant proliferation and metastasis. Despite advances in clinical and basic researches, the molecular mechanisms of serous ovarian cancer progression is still not clear. Recently, many evidence indicates that the molecular mechanisms of cancer progression is not only relevant to the protein coding genes, but also to the non-coding RNAs. Long non-coding RNAs, that is linker, whose length is more than 200 bp and is once thought to be the noise of genetic transcription, but recently has been recognized as the functional molecules. Mounting evidence indicates that the linker can exert key roles in human diseases, especially in cancers. Therefore, elucidating the roles of linkers is indispensable for understanding the molecular biology of cancer progression. Nowadays, the linker HOTAIR has garnered public attention. HOTAIR is located within the HOXC gene cluster on chromosome 12 and encodes a 2.2 kb linker molecule. It is well known because it was originally found to be overexpressed in primary breast tumors and metastases, which had been published in Nature in 2010. Later, its overexpression has been shown to be associated with metastasis and poor prognosis in several other carcinomas, including hepatocellular cancer, colorectal cancer, gastrointestinal stromal tumors, and etc. Recently, another study has garnered attention. It has established the involvement of HOTAIR in the alteration of cell proliferation, cell cycle progression, and apoptosis in pancreatic cancer. Despite these findings, however, nowadays, few studies have linked HOTAIR to ovarian cancer. And although our previous work has linked HOTAIR to ovarian cancer metastasis, which has been published in Gynecologic Oncology in 2014, now little is known about the roles of this gene in ovarian cancer cell proliferation, especially in serous ovarian cancer. So in this study, we will focus on serous ovarian cancer and aim to investigate the expression pattern, clinical significance, and biological function of HOTAIR in serous ovarian cancer progression. Next, we design the research. Well, from the clinical aspect, we want to evaluate its expression pattern, perform the clinical pathological analysis, and prognosis analysis. 2. From the in vitro aspect, we want to investigate the effect of HOTAIR on serous ovarian cancer cell proliferation and clarify the underlying mechanisms of this effect. 3. From the in vivo aspect, we want to construct an in vivo tumor growth model to test the in vivo tumor growth in order to verify the in vitro results. The materials and methods are following. Clinically, we first collect the tissue samples and patient data. We then use qRT-PCR to do the expression value test. Next, we use the chi-square test to do the clinical pathological analysis, and furthermore use the KM method, log rank test, and tox model to do the prognosis analysis. In vitro, we first collect the cell lines and do cell transfection with sRNA. Then we use the MTT assay and plate colony formation assay to do the proliferation test. Next, we use the flow cytometry assay to do the cell cycle and apoptosis analysis. Furthermore, we used the Western blot to test certain dysregulated proteins. In vivo, we first construct the stable knockdown cell line and then construct the in vivo tumor growth model to test the tumor volume, and after the mice were sacrificed, to test the tumor weight. The results are the following. Clinically, 68 serous ovarian cancer and 30 normal ovarian tissues were collected at our hospital between January 2006 and December 2008. From this figure, we can see that using qRT-PCR, HOTAIR levels in serous ovarian cancer tissues were significantly higher than that in normal controls. For the clinical pathological correlation analysis, we first divided the 68 serous ovarian cancer patients into two groups according to the median HOTAIR expression value, that is the low HOTAIR expression group and the high HOTAIR expression group. And then we collect the patient data, including the age, the figure stage, histological grade, residual tumor diameter, CA125 level, and a size. And we found that the elevated HOTAIR expression was correlated with an advanced figure stage and a high histological grade, but not with the age, the residual tumor diameter, the CA125 levels, and the size. Next, for the prognosis analysis, by using the log rank test, we found that patients with high HOTAIR levels had a shorter survival time than patients with the low HOTAIR levels. Furthermore, using the univariate and multivariate analysis, we found that HOTAIR expression, in addition to figure stage and histological grade, was an independent predictor of overall survival. Thus, from the above clinical results, we can conclude that HOTAIR can serve as a predictive biomarker for serous ovarian cancer, and that HOTAIR overexpression may contribute to the malignant progression of serous ovarian cancer. Based on the clinical results, which indicated that HOTAIR is associated with the malignant phenotype of serous ovarian cancer, and given that the unlimited growth is one of the main reasons for the malignant behavior of cancer cells, we want to know whether HOTAIR can affect the proliferation of serous ovarian cancer cells. So we performed the in vitro experiments. Firstly, we detected the basic HOTAIR expression in 6 serous ovarian cancer cell lines, including A2780, HEY, SQV3, OVCA429, OVCA433, and OVCAR3. As shown in the figure A, we found that A2780 and OVCA429 exhibited the highest HOTAIR expression. So, we silenced the expression of HOTAIR in these 2 cell lines with 2 sRNA to examine the effects of HOTAIR on cell proliferation. Using MTT assay, we found that HOTAIR silencing retarded the cell proliferation in both these 2 cell lines. Furthermore, using the plate colony formation assay, we found that HOTAIR knockdown reduced the colony numbers in both these 2 cell lines. These 2 in vitro results indicated that HOTAIR knockdown may inhibit serous ovarian cancer cell proliferation in vitro. Given that the apoptosis and cell cycle arrest are the common reasons for growth inhibition, we next want to know whether apoptosis and cell cycle arrest contribute to cell growth inhibition induced by HOTAIR knockdown. So we next perform the flow cytometry assay. First, using the cell cycle analysis, we found that HOTAIR knockdown decreased the percentage of cells in S phase and increased that in G2M phase in both these 2 cell lines, indicating that HOTAIR knockdown may retard cell cycle progression. Next, using the cell apoptosis analysis, we found that HOTAIR knockdown increased the number of total apoptotic cells in both these 2 cell lines, indicating that HOTAIR knockdown may promote apoptosis in serous ovarian cancer cells. Thus, from the above in vitro results, we can conclude that the proliferation inhibition induced by HOTAIR knockdown is associated with the regulation of the cell cycle, arrest, and cell apoptosis. As the in vitro results indicated that HOTAIR knockdown can inhibit serous ovarian cancer cell proliferation, we next want to know whether HOTAIR can affect the tumor growth in vivo. So we performed the in vivo experiments. Firstly, we chose HOTAIR sRNA-1 to construct a stable HOTAIR knockdown cell line because HOTAIR sRNA-1 exhibited a more potent inhibitory effect than that of HOTAIR sRNA-2. We then injected the normal control and the knockdown cells into nude mice and analyzed the tumor growth. From the figure A, we found that the tumors formed by HOTAIR knockdown cells were smaller in size compared with the controls, and the gap between the sizes of the 2 groups increased with time. Then, after the mice were sacrificed, the tumors formed by HOTAIR knockdown cells were smaller in weight compared with the controls. These 2 in vivo results indicated that HOTAIR contributes to tumor growth in vivo. At last, we performed the preliminary study on molecular mechanisms through which HOTAIR alters serous ovarian cancer cell proliferation. We chose a certain cell cycle and apoptosis-related proteins, including CDC2, CDK2, CDK4, CDK6, cyclin A, cyclin E, cyclin B1, cyclin D1, BCR2, Caspase3, Caspase8, Caspase9, P53, and BRCA1. Using Western blot, we found that the dysregulation of cyclin E, BCR2, Caspase9, Caspase3, and BRCA1 were dysregulated. These results indicated that these dysregulated proteins can partially explain the HOTAIR-dependent effects on serous ovarian cancer cell proliferation. Discussion. HOTAIR, well known for its involvement in metastasis in many forms of cancer, has recently been found to play important roles in cell proliferation, cell cycle progression, and apoptosis in pancreatic cancer. Inspired by these findings, we want to know whether HOTAIR has effect on serous ovarian cancer. In this study, our data highlight the clinical significance of HOTAIR in serous ovarian cancer and suggest that HOTAIR promotes the serous ovarian cancer cell proliferation through the regulation of cell cycle arrest and apoptosis. These results may extend our current knowledge about the function of HOTAIR to include its proliferation promotion effect in serous ovarian cancer. A limitation of the present study was that we didn't study the exact mechanism through which HOTAIR regulated cyclin E, BCL2, Caspase-3, Caspase-9, and BRCA1. So, further studies are required to explore the underlying exact molecular mechanism. In the end, we will make the total conclusion. First, clinically, HOTAIR is overexpressed in serous ovarian cancer tissues compared with the normal ovarian tissues, and that this overexpression correlates with an aggressive tumor phenotype and a poor prognosis. 2. The functional studies suggest that HOTAIR plays a critical role in the control of serous ovarian cancer cell proliferation, both in vitro and in vivo. 3. Mechanically, this role is due, at least in part, to regulation of the cell cycle and apoptosis through certain cell cycle and apoptosis-related proteins. So far, our study is the first to establish that HOTAIR contributes to serous ovarian cancer cell proliferation and may represent a novel biomarker for predicting the poor survival in serous ovarian cancer. So much for the research, and at last we will share some pictures with you. This is our hospital, the part of our hospital, the Obstetrics and Gynecology Hospital of Fudan University. It is named Hong Fangzi because it is all of the building are red. And this is our university. The Fudan University has a long history. This is Shanghai, the Oriental Pearl Tower, the China Pavilion, the Chenhuang Temple, and the Nine-Twist Zigzag Bridge. So welcome all of you to Shanghai, to Fudan. Thank you.
Many thanks for this very nice presentation. I do have a couple of questions, but first the audience, or you? No? You have questions? No. Okay. I have questions which are relatively general, as usual. It's very interesting. This orientation is very interesting, and as we are in chemotherapy, talking about therapy, I'm wondering about the future implications of your studies and results, not only of you but all people working on pattern. And in terms of therapy, there are three main applications. One is prognostics, one is diagnostic, and one is treatment itself. Regarding prognostic, the diagnostic value, what I understood is that HOTAIR is overexpressed mainly in advance of ovarian cancer, already very much identifiable by cytology. Am I correct? It's more the HOTAIR overexpression.
Overexpression
is pretty in late stage cancer.
Yeah.
Did I understand well?
In figure stage 3 and 4.
Stage 3 and 4.
Yeah.
So it's stages where diagnosis is already relatively, in brackets, easy with other methods. Am I correct?
The figures and the histological type.
It's already very well identified.
Yeah, it's very, very important in these ingredients.
Okay, so there is relatively less interesting in terms of diagnostic as itself. Now, if we talk about diagnostic, I understood that what you showed is that in patients, it's a prognostic. Overexpression of HOTAIR in ovarian cancer is a prognostic of earlier death. So there's two figures, 2 lines.
Yes.
Yeah.
When should the Kaplan-Meier occur?
Yeah, Kaplan-Meier occurs.
Yeah.
Yeah. Here. And it's pretty striking. There is a huge difference. I don't know the numbers.
However, the numbers of the patients are small. It is 6 to 8. If later we will augment, make augment of the patient data. We will at least 200 people is.
Okay. What I'm wondering is in terms of, as I mentioned, diagnostic.
This is the limitation.
Yeah, I understand it's in patients. But this could be interesting in order to select either patients with a high potential of mortality or select patients with a low potential of mortality when we are developing new drugs. What I mean by that is that currently, if you are developing a drug for ovarian cancer, for instance, you are taking both populations mixed, and it may be because you don't know that in one of the in the control group you have more high HOTAIR plus patients, and in your active group you have HOTAIR more minus HOTAIR minus patients, meaning that even if your drug is efficient, it becomes non-efficient in the end because you have enrolled more patients with a bad prognosis at the beginning. So it could be useful for that drug development. The other one aspect is also maybe your drug is could be interesting to select patients with a higher risk of death if your drug is potent because it's going faster, and a smaller difference will be significant than if you are using selecting only HOTAIR minus patients. So it could be very interesting in terms of diagnostic, I guess. And also in the other sense, maybe HOTAIR minus could be more sensitive to one drug than to another. So from my just speaking like this, is that could be a very good news, very good diagnostics tool, very, very good one. So it's very interesting in that sense. So it's more a feeling than a question. But do you know if some activities are made towards diagnostics with that, or if you are thinking of that, or maybe you may think of that in the future? We were just mentioning that you will increase the number of patients here, which is good. But at some point, maybe you combine that with sensitivities to different types of anti-cancer agents? Do you know if it is done? If it is done that some teams test the response to existing anti-cancer or future anti-cancer agents and take as a variable the HOTAIR expression?
We didn't include this to this research.
No, you not, but do you know if in the community, in the world, you may not know, I don't know, that's just, no, you don't think so, because it might be interesting. And actually it's coming to the third aspect, which is the treatment. Well, you have made knockout mice, but you cannot make knockout women. It's a bit difficult. You may have maybe anti-RNA treatments could be of interest, but that's for the time being it doesn't work very well. Are you going into? Well, you have begun to look into the expressions of the caspases and other things that are more important in HOTAIR++ patients than in HOTAIR negative patients or mice. I don't know. Is one of your goals or the goal of the community also to identify mechanisms that are or targets, drug targets that could be overexpressed or over-functional in HOTAIR++ patients? If you don't understand, ask me to rephrase. I can rephrase because it's a bit complex, the question. No? Okay. You understand what I mean? Maybe you can express it.
Your test could be used by different companies doing different studies. First of all, to have a homogeneous group of people, the people who are tagged to die or to survive longer. And probably also the Food and Drug Administration or other agencies could ask the company doing a study with a new drug to select the patient based on the study. And if you have a very nice drug, very potent, then you may choose those patients who are already tagged to live shorter. Or if you have a drug which is not so good but is good, you may choose the other, but not to mix them. Okay? So it's like a commercial test. If you put a commercial test, you may could be used to see, okay, I have this cancer in the third stage. He has a very low HOTAIR expression, so he will live longer. So you will use different strategies. Or if you know that he will live not so longer because the expression is very high, you'll be more aggressive, or you use another new drugs and so on. So you should commercialize the test to be put like a pregnancy test, let's say.
Yeah, or
chromatographic.
There is patent protection and everything.
Probably you did it, you know. Someone in Shanghai. Yeah.
Sorry, I just wanted to find out about your methodology. How did you get the normal samples? Normal ovarian samples, are those volunteers?
From the ovarian cyst or the ovarian cyst, the ovarian cyst,
from the cysts?
Yeah, cyst, the normal ovarian tissues.
That removed from women?
Yeah.
Okay. So it's not just that they're normal, it's just that the cysts are not cancerous?
Yeah.
Okay. That's because I wanted to know how that was gotten.
I don't think it's healthy volunteers. Yes.
And do you evaluate also the disease-free survival, or do you follow up the response of these patients to treatment? Because maybe if there is a recurrence of the disease, you could use this as a marker of treatment follow-up, or a marker of recurrence. It could be a marker. Have you checked the recurrence of the disease or the disease-free survival?
She doesn't understand that somebody is.
Can you speak louder?
Sorry. Do you evaluate these patients after time to response to treatment, like if the disease recurrence or if they responded to treatment specifically, not death, but response to treatment?
We haven't included the response to treatment.
What she's asking is about follow-up. Am I correct?
Yeah.
Follow-up after your treatment.
It's just a curiosity because maybe this HOTAIR could also be a marker of following up the patient response to treatment.
Yeah, this is a very good comment.
They look only at the survival, no matter what the treatment the patient received. But as you say now, knowing that the higher expression means lower survival,
Yeah, maybe
it could be applied.
maybe patients in low stage could present this higher expression, and it could be a marker of evolution of disease, a progression of disease, maybe.
Yeah.
So have you ever tried introduction of HOTAIR expression vector into the low expression cancer or knockdown cells, and to see whether the overexpression, artificial overexpression of HOTAIR can induce high malignancy?
I just divided the high and low.
Yes, I know. But have you ever looked at effect of artificial HOTAIR overexpression on the malignancy of cells which are knocked down by cytokine?
Yeah, we knocked down by some.
Yeah,
but
so you can see the cell cycle arise and up.
I haven't do the expression experiments. This is also the limitation. Thank you.
It was my understanding that the volume of the tumor changes in relation to the expression of HOTAIR.
Yes.
Now, when you inject with HOTAIR, the volume of the tumor
The volume?
You show the volume of the tumor
Yeah.
and the effect of HOTAIR. And you show the volume is larger or shorter.
Also.
Yeah, you can show in the lab.
Okay.
Yeah.
But we
You
didn't do the. We should.
Yeah, tumor volume.
In figure C.
And days after the injection.
In figure A.
This is the cells are already knocked down, so the cell cycle is retarded. So it's great. So these cells, before injection, these cells are already treated with siRNA.
What she said that we found that tumors formed by HOTAIR, knockdown cells were small.
Yeah.
So you do not, if you do not have HOTAIR, the volume is small.
Yeah, but the growth is also small.
Yeah. And the growth probably is smaller because they stopped. It stopped apoptosis.
Induced
So induces apoptosis. Tumor cell apoptosis.
Yeah, I understood that Dr. at the very beginning of the presentation you were talking about chromatin, I think? No? Yeah.
It was the chromatin and the acetylase inhibitors.
Yeah. Well, whatever.
Yeah.
Okay. I don't know if there are other questions or if you have other comments. Unfortunately, sorry. The conference as for you. Thank you very much for the talk.